Thanks so much for joining me. My name is Max Sellman. I'm the product manager for gene editing here at Aldevron. I'm here to talk to you today about CRISPR-associated nucleases for gene editing, tools to support discovery and therapeutic programs. First, by way of backstory and introduction, just to tell you a little bit about Aldevron, we've been around for over 20 years. We're a manufacturing company. You could call us a CDMO. We're manufacturing plasmid DNA, mRNA, and proteins from research grade through GMP. We're supporting clients from early discovery all the way through commercial GMP applications. We also have an antibody discovery unit that does novel antibody generation using traditional immunization as well as a proprietary genetic immunization platform. In a nutshell, our mission is simple. Our clients are out here coming up with revolutionary new approaches to all manner of medical challenges facing the world: cell and gene therapy, diagnostics, drugs, etc. And for Aldevron, our mission is to be the top quality raw materials supplier. Our CEO Mike Chambers likes to say, if you're going to build a railroad, you need picks and shovels. And so for Aldevron, our goal is to provide the highest possible quality picks and shovels so that our clients can build an excellent railroad. So I'm here today to talk about CRISPR nucleases. This is something that Aldevron has been involved in for a number of years now. We were the first company to launch a GMP Cas9 as a catalog product. And it's something we still do today. And so the idea is that you can come in to work with Aldevron when you're still pretty early in your product development life cycle. You're still working on your process. You're still in the lab. You need research grade product. And then when the time comes to actually translate, to move out of the lab into the clinic, to start working on treating patients, that you would have access to precisely the same nuclease that you were using for your development, manufactured to the same standards, released with the same quality specifications, and with the same expected performance. Not better, not worse, the same. So the idea is that you don't lose valuable time, waste valuable effort, re-optimizing your process after you've developed it. You can go directly in to treating using gene editing in a clinical workflow, and you can do that with immediate access to a nuclease that you don't have to manufacture custom. You just buy it off the shelf. It's available right away. This has been a really popular strategy for CRISPR nucleases and has really allowed Aldevron to take steps to really lead the field in the development, the implementation of quality control specifications, assays, and new nucleases entirely for the cell and gene therapy markets. And so we wanted to spend a little bit of time today talking through some of the good work that our team has done to generate these robust analytical methods for gene editing nucleases, and then to talk a little about how they've been applied to some pretty exciting nucleases. So to give you the proper context here, I think we first need to have an understanding of what sort of quality control specifications we're talking about. Aldevron, for our gene editing products, and this panel is true for all of the research grade nucleases we offer, as well as all of the GMP nucleases that we offer. Consistency. We've got 13 different tests. There aren't any other suppliers that are doing this deep of analytics into their CRISPR nucleases. And for us, the big questions were to make sure that we're manufacturing a nuclease that is of high quality. It's pure, it edits well, it is of a high concentration, it has good product characteristics, but it also is free of critical contaminants. So you want to have undetectable levels of residual nuclease. You want endotoxin as low as possible. You don't want there to be bioburden. So these were all the kind of philosophies that we brought in as we started to develop this QC test panel, which is what can we test for to make sure that we're checking all the boxes that our clients need to check to ensure that they have a suitable material for use eventually in a clinical environment. Very briefly, just to show you the actual specifications that we use. just to point out that they are very tight specifications. The idea, again, is to produce a protein of the highest quality. Um, so the first thing you'd want to understand, if you're in the business of manufacturing Cas9 and selling it to clinical clients, is to make sure that the product that you're making is made in a reproducible manner. That is to say that if a client sources some Cas9 to use today, and then they source a different lot of the same product to use, let's say, in a month or in a year, our clients want to be sure that the performance is going to be equivalent from lot to lot. They're not going to have variation between lots that is going to cause undue problems in the execution of their experiments. And so what you see here are a few overlays, us taking a look at specific activity in vitro, looking at HPLC purity, and then looking for the presence of aggregates via analytical size exclusion chromatography. And what you see when we overlay, these are just 3 example lots, but you could actually do this with all of the lots of this protein that we've made. You see almost complete overlap of these lines. It's giving us a strong indication that the proteins that we're making are performing very equivalently and have the same profiles time and time again, month over month, year over year, so clients can be assured in the long run of the performance of this product. And just some interesting data that you can take a look at when you go a step forward here is to actually take the Cas9 and apply it to, in this case, T cells, specifically a TCR alpha gene in T cells. This data was generated by our collaborator at University of Minnesota, Mark Osborne, and it was just a nice little experiment to demonstrate that when you target Aldevron Cas9 in vivo at a therapeutically relevant target, you do get very suitable editing. And so you don't necessarily always see that when you buy a commercially available Cas9, but coming in with the goal of producing a product of clinical quality, it was good to see that in this application, the nuclease was suitable. Now, another aspect to consider with analytics is that it's great to have a deep set of QC tests performed on your nucleases, but you do need to be sure that those QC tests are going to actually detect the presence of some sort of product that is declining in quality. So what we wanted to do was what we called a forced degradation study, where we took our SP-Cas9 product and we put it through a number of adulterating conditions, the whole goal being to create low quality product that could be detected and characterized by these assays. And so on the top of this slide, what you're seeing is a look at concentration. On the bottom, you're looking at activity as we adulterate with the addition of a peroxide or an acid to lower the pH or an increased temperature. And what we see on this slide is that quite clearly we can detect evidence of a drop in concentration, a drop in editing efficiency. And when we look at our some other assays, such as HPLC for purity, well, if we digest the protein with trypsin, cut it up into a bunch of pieces, we can see the degradants and the kind of pattern in which Cas9 typically degrades in this experiment. It's very easily detectable. We can also detect common aggregates over a period of time when the product is treated with peroxide. The bottom line here being that through a series of experimentations and development, we've been able to make ourselves highly confident that we're able to detect when our Cas9 is no longer meeting the high quality specifications that we've set for it. One other interesting point that we discovered in this process, and we're able to bear out with some data, is that Cas9 is quite stable as a protein, but it is a protein. So when you heat it up to upwards of 40 degrees, it doesn't like that. That diminishes its activity. We did find, interestingly, though, that a way to protect that activity is to complex the Cas9 into the RNP. So combine your Cas9 with the guide RNA into a ribonucleoprotein complex. when we did that, we actually found using a thermal shift assay, as well as some of these other techniques, that the complexed product was much more thermally stable, and maintained its editing efficiency over a larger window than unbound Cas9 that had been heat treated and then bound and then targeted. So we just thought that that was some interesting data to share. Um, Lastly, as it relates to quality and stability, these assays, we actually perform them over a period of months in a stability study on our products to ensure that this protein would maintain its quality over a period of time. And in the case of the research grade Cas9, we actually found that our formulation is stable for more than 2 years. We took the study out to 30 months and did not see any degradation of the product as it relates to the assays that we were performing. So out over 2 years, we saw consistent high purity, high editing efficiency. This just kind of really drives home the key points that the protein is fairly robust, and that the manufacturing method that Aldebaran is employing is consistent and isn't leaving behind anything in the solution that is causing it to degrade quickly. Those are good pieces of information to know. Here's another look at that data, another way to look at the purity via SDS page, as well as the concentration remaining remarkably stable over time. So finally, in the last couple of minutes here, I would just like to briefly turn towards what a product that we're very excited about here at Aldevron, which we call SpyFi Cas9 Nuclease. And a great place to start learning about this is actually in the paper where IDT, in collaboration with Matt Porteous's lab at Stanford, first announced the discovery of this variant, which is a high fidelity nuclease, one single point mutation from an arginine to an alanine, causes off-target editing from wild-type Cas9 to virtually disappear. Now, at the same time, it doesn't really impact the on-targeting editing. So you can imagine if you're a researcher who has interest in bringing Cas9 into the clinic, and you're concerned about off-targets, maybe your gene of interest, your guide of interest has a specific known off-target profile that is causing you concern, it would be great to learn that there is a nuclease out there that can virtually eliminate off-target effects. And so just taking a brief look at that data, what you've got here in the blue bars is Aldevron wild-type Cas9. We also included in gray just a competitor product, just to give another example of a wild-type Cas9 in action. And then in the kind of orange and red warm colors, we've got the Aldevron SpyFi Cas9, which is produced under license and in partnership with IDT. And what you're seeing here on the left is good on-target editing efficiencies for all of these nucleases, especially at higher concentrations. You're seeing near complete editing on target at the point of interest. Now contrast that though with your off-target sites. What we did in this experiment is we targeted with a guide that we knew with wild-type Cas9 would cut off-target at a number of sites. And the question that we're asking with this experiment is, can we reduce off-target effects by using SpyFi instead? And the answer is a fairly resounding yes. And this is just kind of an image showing one such experiment, but we've seen this borne out dozens of times over with various clients in industry and academia who are concerned about off-target effects, switch from a wild-type SP-Cas9 product to Aldevron SpyFi for clinical purpose, and they see those off-targets go away. And so we're really excited to be able to partner with IDT to provide this from research through GMP as an off-the-shelf product. It I think really speaks to the power of this nuclease that 3 years after its discovery, this is still absolutely hands down the industry leader, the field leader for a reduced off-target editing Cas9. There have been a lot of them that have been discovered and published, but this is the one that's really taken. And I think that really just reinforces the strength, the power of this nuclease. And so I really appreciate you taking the time listening to this talk. If there's any questions that you have about the content of this or really anything related to Aldevron CRISPR nuclease products, I would encourage you to reach out to me. My phone number, as well as my email address, are available on the screen here, and I should be reachable there as well as throughout the normal kind of modes of communication at a conference. So thanks so much, and take care.